Hello, my name is Milton Jennings. I have a 4.2 GPA and I have a 1400 super scored SAT score, and I attend Cass Technical High School. I am also a student science at Ecotek Labs, and my preferred career goal is to become a neurosurgeon. Today, I'm going to walk you guys through the autoclaving process and how it presents bacterial growth on surgical equipment. 2 summers ago, I did cancer research in Wayne State's pharmaceutical building with Dewey Long and Dr. Ayer's lab. We were trying to figure out how to deliver CD40 antibody to a tumor's microenvironment without CD40A getting leaked into the entire body. We wanted to do this because CD40A is toxic to the entire body, so we decided to use liposomes, which are these microscopic discs that can carry the CD40A and deliver it to the tumor's microenvironment. These liposomes knew when to release the drug because of the tumor's different pH than the rest of the body. So the thing we did was make the liposomes using lipids L-A-phosphatidylserine and DSPS PEC 2000 DMA, and the thin film method, which is the method used to combine these two lipids and the CD40A drug into a liposome. Once we configured the liposome, we tested to see if it was uniform using a dynamic light scattering machine. After we figured out the liposome was uniformed, we used a dialysis device to see how fast the CD40A leaked into the different pHs. And our results suggested that negligible amounts of CD40A releases into the body when delivered to the tumor's microenvironment, and CD40A releases much faster in the tumor's microenvironment. So along with the cancer research, I interned with Dr. Entz, an internal medicine doctor at Henry Ford Hospital. While interning, she told me about how hospitals use autoclaving in order to sterilize surgical equipment. And upon further investigation, I found that autoclaving typically uses temperatures of 121 to 134 degrees Celsius, achieved by pressurizing steam to ensure the complete destruction of all microorganisms. At these temperatures, sterilization usually takes around 15 to 30 minutes, providing a thorough, reliable method for infection control. And before surgery, instruments are cleaned and placed in an autoclave, where high-pressure steam sterilizes them to kill bacteria, viruses, and spores. So during surgery, only sterilized instruments are used to maintain a sterile field and reduce infection risks. And after surgery, any reusable tools are cleaned and autoclaved again to prepare them for the next procedure. If instruments aren't autoclaved, harmful bacteria, viruses, or spores can survive on their surfaces, leading to a high risk of infection for the patient. This contamination can result in complications, prolonged recovery, or serious, sometimes life-threatening infections. Now we're back to the lab, and I'm basically going to show you guys how to autoclave. So right here we have a continuous stitch that I did in order to close this enclosure up. As you can see, it's now closed, and you can also see these others that aren't. And so what I did in order to do this was these 4 instruments. So right here we have an iris scissors, which were used to cut the suture string. And then we have clamps, which I used to hold the metal that I did to do the sutures. And then we have tissue forceps and actin forceps, which are commonly used in surgeries. And so basically, if these tools aren't autoclaved, then there's a higher chance of infection. And so now I'm gonna show you guys how to make sure that higher chance isn't reality. And before I show you guys the autoclaving process, this is a picture of bacteria that can grow on surgical equipment. And definitely wouldn't want that anywhere near someone's body. Now I'm gonna take you through the steps of autoclaving. So the first thing that you wanna do is plug it in. I'm gonna do that here. And so the next thing that you wanna do is open this lid, take these metal rings out, and put your surgical equipment in here. So once it's spaced out, so you don't really want them touching each other, that can affect it. And so once that's done, most equipment can get fully sterilized at 125 degrees Celsius after 15 to 30 minutes. So we're going to do it for 125 degrees Celsius. Would you take this knob here? And so it's 10 intervals. So you want to get to 120 right about there and move it a bit more. And so then that's 125. And then you want to set it to 15 minutes, which you can set right there. And so once that's done, now we wait for the 15 minutes to be up for it to be fully sterilized. Okay, so now that we heard the ding, that means it's done. So once we're done, open this lid, take these rings, hook it right here, and take out the equipment. Now, because the equipment did just get heated at 121 degrees Celsius, it's very, very hot, so you have to wait for it to cool down. But while we wait for it to cool down, I'm gonna discuss how the autoclaving experience kills cells. So basically, once the machine gets to 121 degrees Celsius, the bacteria the bacteria cells are affected by this. But how it's affected by this is the heat basically gets into the cell, and once that happens, there's protein chains. And so the heat disrupts these protein chains and ends up, the cell won't be able to produce protein. And once that happens, the cell basically ends up dying. And so that's how it kills the bacteria cell. And so now back to this. So now that these should be cooled down, yep, you take these, put it on a sterile platform, and we already sterilized this beforehand. And so now we have sterilized equipment. And that's pretty much the autoclaving process. I hope you learned more about the process of sterilizing surgical equipment using the autoclaving process. My time at Ecotek Labs has given me the tools that allow me to dive deeper into the science behind things, and it allows me to turn my ideas into measurable actions.